Hello, my name is William Bernfeld from King School CT, and today I will be presenting research on the structural determination of novel non-canonical base pairs and mismatches. Over the last 2 years, the novel coronavirus has claimed over 4.5 million lives. Though preventative treatments such as vaccines have proven highly effective against the virus, direct treatments for COVID-19, while in development, have not been released to the public. In response to the demand for such a direct treatment, we've developed 4 novel non-canonical base pairs. Before we could design the base pairs in question, we had to understand how DNA self-assembles and organizes itself. Through a set of molecules known as nucleobases, a given strand of DNA forms hydrogen bonds with an adjacent strand, forming a stable double helix. In order to do this, a given nucleobase possesses functional groups that act either as hydrogen bond donors, shown in blue, or hydrogen bond acceptors, shown in red. Through the alignment of bond donors and acceptors and the hydrogen bonds they form, shown in green, the 4 nucleobases found in canonical or natural DNA form a set of 2 base pairs, one between cytosine and guanine, shown here, and one between thymine and adenine, shown below. As seen in this visual, cytosine and guanine share a set of 3 hydrogen bonds, making them thermally more stable than the thymine-adenine counterpart, which only has 2 hydrogen bonds. Though life as we know it is fully functional using the 4 nucleobase language of canonical DNA, there are limitations to this layout. For one thing, the presence of only 2 base pairs offers little sequence diversity, and therefore allows only a select few amino acids to be encoded. In the case of eukaryotic organisms, only 20 out of hundreds found in nature. In recent years, however, scientists have expanded upon this 4 nucleobase twofold, in the form of a language known as Hachimoji DNA. In this model, the 4 canonical nucleobases are accompanied by 4 synthetic analogs, which subsequently assemble into 2 additional base pairs, each with their own set of 3 hydrogen bonds. As a result of the stability of Hachimoji DNA and the nucleobase it contains, it can be transcribed into RNA and therefore can encode novel polypeptides. While the 4 base pair language of Hachimoji DNA offers a more versatile, diverse genetic code, it too is limited by relying solely on hydrogen bonds to link complementary nucleobases. To combat the novel coronavirus with the greatest impact, direct treatments must rely on more unique methods of base pair formation. To bypass the obstacle of hydrogen bonding, in which each nucleobase must have a single complement, my mentor and I have employed a novel model known as metal-mediated DNA, or mmDNA for short. Rather than using hydrogen bonds or other intermolecular forces, mmDNA forms base pairs between formerly incompatible pyrimidines, which, as shown in this visual, cannot form hydrogen bonds with one another on account of having no complementary functional groups. In lieu of this, we use transition metal ions, such as the mercury 2+ ion seen here, to form coordinate covalent bonds, shown in green, with hydrogen bond acceptors, shown in red, as ligands. With bonding strength that surpasses intermolecular forces, mmDNA offers further stability and resilience to sequences that contain metal-mediated mismatches. In addition to mmDNA, we employ a semi-canonical nucleobase analog, named as such because it is only found in the genetic code of certain bacteriophage viruses. The nucleobase in question, 2,6-diaminopurine, or DAP for short, is nearly identical to adenine, its closest counterpart in nature, the only difference being the presence of a second amino group, allowing it to form 3 hydrogen bonds with thymine or a given uracil derivative, offering increased stability to DNA sequences that contain DAP. With these concepts in mind, we present a set of 2 base pairs containing DAP, one between DAP and thymine, a bird's eye view of which can be seen here, a side view of which can be seen below, and one between DAP and 5-iodouracil. 5-iodouracil is nearly identical to thymine, the only difference being the presence of an iodine atom connected to the 5' carbon instead of a methyl group. Moreover, we present 2 metal-mediated base pairs, a pair of mismatched thymine bases mediated by a mercury 2+ ion, and a mismatch of cytosine and its isomer, isocytosine, which is featured in Hachimoji DNA. This base pair is mediated by a silver+ ion. The cytosine-isocytosine base pair, shown here, was the focus of my project last year, and was designed by my mentor. However, I have now redesigned the base pair so as to remodel it as a metal-mediated mismatch, which uses coordination covalent bonds in lieu of hydrogen bonds. Before we could synthesize the base pairs in vitro, we had to make sure that our ideas were practical, let alone possible. To test the theoretical stability of our base pairs, we ran a set of simulations to analyze their hypothetical structure. To begin this, we downloaded a digital model of a pre-existing motif from the RCSB protein databank. We then used a variety of softwares to modify the base pair at the center of the DNA sequence so that it reflected one of our 4 base pairs, ultimately producing a set of 4 modified motifs. The standard laboratory procedure for analyzing novel base pairs is through X-ray diffraction, which gives us an accurate representation of how the pair fits into a larger sequence. To make this possible, we had to design a DNA sequence that could self-assemble into a crystal we could then diffract. To do this, we employed a method known as sticky-ended cohesion, in which single-stranded extensions, an example of which is shown here, is made to a DNA double helix, which subsequently forms hydrogen bond with a given complementary sequence. For example, the sticky end sequence ATGC would only form hydrogen bonds with the sticky end complement TACG. Therefore, identical DNA sequences with complementary sticky ends can subsequently self-assemble into supramolecular structures, including, but not limited to, the motif we use to produce our crystals. The motif we used is the tensegrity triangle, which is made from 3 identical strands of DNA. The sticky ends of a given sequence protrude from the tensegrity triangle it occupies, allowing individual triangles to self-assemble into rhombohedral crystals, a representation of which can be seen here. Using the software Phoenix to simulate the self-assembly of our crystals into crystalline lattices, we compared their structure with experimental data from the motif we used as a template. This data, known as an electron density map, or EDM for short, is a representation of the electron cloud surrounding a given molecule, the shape of which is determined by the scattering of X-rays during diffraction of a crystal structure. Once simulated self-assembly had finished, we compared the pre-existing EDM, shown here as a blue mesh, with the theoretical coordinates of our modified motifs, shown here. Because the EDM and molecular coordinates overlapped, we inferred that the motifs we intended to use would indeed form a crystal similar to the motif that inspired our design. To synthesize our base pairs in vitro, we used phosphoramidite-based oligonucleotide synthesis to assemble individual DNA strands, which then self-assembled into double helices over the course of the next 12 hours. To allow our mmDNA base pairs to form, we suspended the strands containing mismatched bases in solutions of 0.001 molarity mercury-2 nitrate and 0.001 molarity silver nitrate for the thymine-thymine mismatch and cytosine-isocytosine mismatch, respectively. After self-assembly, we purified the sequences via agar gel electrophoresis, separating the purified sequences from the gel via a 2 micrometer centrifugal filter, using repeated doses of heat to anneal them over the next 24 hours, so as to facilitate self-assembly into rhombohedral crystals. After the 24-hour period had elapsed, we shipped the crystals off to Argonne National Laboratory, where we would conduct diffraction. Because of COVID-19 related restrictions, we conducted our analysis from a distance, using a remotely controlled robotic arm to place each crystal within a diffractometer, conducting extra diffraction afterwards. Our simulations thankfully yielded stable electron density maps, such as the detailed double helical EDM seen here. The overlapping of the EDM with the coordinates we ascertained from diffraction suggested that the crystals were stable upon diffraction, and that sticky-ended cohesion, a model of which can be seen here, did indeed take place. Through post-diffraction analysis, we were able to process the data so as to produce detailed images of tensegrity triangles, such as that shown here, and the rhombohedral crystals they self-assembled into, such as seen in these visuals. In both our simulated and experimental datasets for the thymine-thymine mismatch, the distribution of X-rays is roughly logarithmic as the size of the crystal increases, indicating structural uniformity. The agreement between these two datasets, shown here as an R factor, reaches near zero at a resolution of just over 3.16 angstroms. Scattering of diffracted X-rays reaches a peak at a resolution of roughly 3.73 angstroms, indicating, through a precise resolution, that our crystals were structurally uniform. Because this resolution was so precise, we have reason to believe that the crystals did indeed form and that little impurities or anomalies were present. The same trends can be observed in the data for our cytosine-isocytosine mismatch, our DAP-thymine base pair, and our DAP5 iodouracil base pair. The results of our data suggest the stability of our 4 base pairs, implying that they can both be modeled in silico and then efficiently replicated in vitro. Moreover, the accuracy of our simulation suggests that we may be able to use a similar procedure for modeling future base pairs if need be. Through the use of metal-mediated mismatches, we present the possibility for a new kind of hybridization probe used to neutralize genomic viral RNA. The coordinate covalent bonds formed between mismatches, a probe containing our mismatched base pairs may render coronaviral RNA inseparable from its adjacent strand, and therefore unreadable by cellular machinery. As a result, it may neutralize the novel coronavirus before it has a chance to spread further throughout the body and perhaps even kill someone. Additionally, the use of DAP may provide tighter binding hybridization probes by which we may track the propagation of the novel coronavirus in vivo. Additionally, the use of rhombohedral DNA crystals, such as those used in this investigation, may offer possibilities for controlled drug delivery, as the nanocage-like structure of our crystals makes it an ideal candidate for the precise arrangement of novel drugs in vitro, as well as their intentional release upon introduction to a living system. Our investigation also opens the door to the encoding of non-canonical amino acids. that is, amino acids which are not encoded by naturally occurring genetic material. Through the use of DAP and novel base pairs, we offer the possibility of expanding the genetic code beyond the 20 standard amino acids found in eukaryotic cells. With this increased diversity of monomers, we may produce oligopeptide and polypeptide drugs capable of fighting the novel coronavirus with extreme efficiency. With an expanded genetic code, we may produce secondary, tertiary, and quaternary protein structures that would not be possible without non-canonical amino acids, such as synthetic antibodies used to neutralize COVID-19 spike proteins. With our novel base pairs and their applications in future antiviral technologies, we hope to not only change how we look at COVID-19, but how we treat viral infections as a whole. While viral outbreaks are, of course, impossible to prevent entirely, the structures we have developed may very well act as tools by which a future viral strain may be treated before it proliferates to the catastrophic effect of a global pandemic. I would like to thank the following people for their mentorship, guidance, and support over the course of this investigation. Without them, I would not have the incredible opportunity of taking part in this project, nor would I have the honor of having my research be featured in an upcoming Nature publication. I would also like to thank you all for taking the time to listen to my presentation. I am now open to questions.